Hello students, in this lecture we will study the uses of biodiversity, the threats to biodiversity and the conservation of biodiversity by the means of this presentation. The word biodiversity is a contraction of biological diversity. The father of biodiversity, Edward O. Wilson, an eminent entomologist, first coined this term in 1986. Diversity is a vast concept, refers to the range of variations or differences among some set of entities. Biological diversity thus refers to variety within the living world. The diversity can be of any type, maybe the morphological pattern of the human being, maybe the eating pattern, maybe the variety in the animals, maybe the variety in the dressing pattern, maybe the variety or diversification in the traditional they were following. So over here, we are concerned about the diversity in plants and animals. The term biodiversity is generally considered as an umbrella term referring to organisms found within the living world. It is commonly used to describe the number, variety of life, and variability of living organisms. Biodiversity, the variability among living organisms from all sources, including inter alia, among other things, terrestrial, marine, and other ecosystem, including the aquatic ecosystem, and the ecological complexes of which they are part. This include diversity within the species, between the two species of ecosystem. Then comes the elements of biodiversity. The predicted number of total species varies from 5 to 50 million and averages at 40 billion. Out of the total known species, about 60% are insects, about 16% are higher plants, and only about 0.3% is mammals. The most unique feature of Earth is existence of life, and the most extraordinary feature of life is diversity. Biodiversity is normally treated in terms of genes. It can be in terms of species, and it can be on terms of ecosystem. You can see the biodiversity in the genes. Then the biodiversity among the species or within the species also you can see the biodiversity, and you can see the biodiversity in the ecosystem. There are various types of ecosystem we have studied in previous lectures. In correspondence with three fundamental hierarchical levels of biological organization. The three diversities are referred as the genetic diversity, species diversity, and ecosystem diversity. Sometimes landscape is a heterogeneous land area composed of cluster of interacting ecosystem. that is repeated in similar form throughout or mosaic of heterogeneous land form. Vegetation types and land uses or pattern diversity is considered as fourth form of biodiversity. Diversity can be seen within the species is genetic diversity. Between species is species diversity, or we can call it as taxonomic diversity or organismal diversity. At ecological or habitat level is ecosystem or ecological diversity. So over here in diagram we can see the types of diversity. The genetic diversity is concerned about the differences in DNA among individuals. Species diversity in that variety of species in a given area. So whatever the species which are in a particular area comes under the species diversity. Then comes the ecosystem diversity, variety of habitats, ecosystem communities. Interaction is the principal intrinsic mechanism that shapes the characteristic and function of biodiversity. These interactions are a hierarchical nature, hierarchical level of biodiversity and their interrelationship. In that, if you see the genetic diversity, species diversity, and ecosystem diversity, they are in an interrelationship with each other. Means if one of the diversity will be altered, then obviously it has an impact on other two diversity. If there is a diversity in the ecosystem, then obviously it creates the species diversity or it creates the genetic diversity. Then the scale of organization. Genetic diversity, it starts from the molecular level, then comes to the cell level, then comes to the individual organism, then comes to the population, and then comes to the community. And resultantly, it gives the genetic diversity. Then, if we go for the species diversity, then the base for that, that is a subspecies. From that, you can see the species. From a species, it moves to the genes. From genes, it moves to the family. From family, it moves to the order. From order, it moves to the class. From class, it moves to the phylum. And from phylum, it moves to the kingdom. For ecological diversity, it starts with the habitat, then move for the patch, then move for the ecosystem, then move for the biome, and then move for the biosphere. And directly, it has an impact on the ecological diversity. The scale of organization and level of interaction of biodiversity, that how each and every level impacts or create the biodiversity to its next level. Genetic diversity. Genetic diversity referred as the diversity within the species or intra or infra species diversity. It encompasses the components of genetic coding that structures organisms, include nucleotides, genes, and chromosomes, and variation in the genetic makeup between individuals within a population and between population. The variation could be In alleles, different variants of same gene. In entire gene, the traits determining particular characteristics, or in chromosomal structures. This enables a population to adapt to its environment and to respond to natural selection. If a species has more genetic diversity, it can adapt better to the changed environmental condition. Lower diversity in a species leads to uniformity and the amount of genetic variations in the basis of speciation evolution of new species. It has a key role in the maintenance of diversity at species level. Genetic diversity within a species often increases with environmental variability. It can be measured by the methods based on DNA marker like RFLP, PCR, RAPD, APPCR, and PFGE, microsatellite, prime PCR, and others. Species diversity. Species are distinct units of diversity, each playing a specific role in an ecosystem. Therefore, loss of a species has consequences of the ecosystem as a whole. So, if there is a loss of a species, if the species is toward the endangered, means somehow it is a consequence of the ecosystem, it is a consequence of change in the ecosystem due to any of the factor. Refers to the variety of living species within a geographical area. It encompasses the taxonomic hierarchy and its components from individuals upward to species, genera, and beyond. The simplest measures of species diversity, SR plus SC, are the first, that is the species richness, the number of species per unit area. So, if a particular species or a number of that species is much more in a particular area or a per unit area, it comes under that how much rich that species in that area. So that comes under the species richness. Then species evenness. The evenness in the number of individual of each species in an area. That comes under the species evenness. Generally, greater the species richness, greater is the species diversity. So, if within a per unit area, the number of species will be more, if the species richness will be more, then we can say obviously the number of the species are more, then we can see the diversity at a very higher level. Alpha diversity. Alpha diversity refers to the average species diversity within a particular area, habitat, community, or ecosystem. It is measured by counting the number of taxa, distinct group of animals within the ecosystem. Beta diversity is species diversity between ecosystem, refers to the ratio between alpha diversity and regional diversity, or comparison of taxa that are unique to each of the ecosystem. Beta diversity. Number of species in habitat 1. Number of species in habitat 2 and 1 plus number of species in H2, that is number of common species habitat 1 and 2. Gamma diversity is the overall diversity for different ecosystem within a region or total diversity over a large area of region. Now comes the uses of biodiversity. The different types of the direct use value of biodiversity are for food, medicine, biological control, industrial materials, recreational, harvesting, and ecotourism. Food in the form of vegetable, fruit, nuts, meat, milk, as well as food colorants, flavorants, and preservatives. For medicine, willow tree, salicylic acid, aspirin, foxglove, that is GD toxin, atropha, belladonna, atropine, opium sativum, codeine, papaver somniferum, morphine, cinchona, legerina, quinine. So these are the medicines which we can get from the nature. Animal source products, example, anticoagulants, coagulants, vasodilator agents, and for models on which the test potentiality useful drugs or techniques. Biological control, that is vector control using biological agents. Industrial materials, building materials, fibers, dyes, resins, gums, adhesives, rubber, oil, and waxes. Agricultural chemicals include pesticides and perfumes. Recreational harvesting is the harvesting of animals. Example, fish, reptiles, birds, mammals for display and as pets. Threats to biodiversity. The global change in the factors responsible for biodiversity results into loss of biodiversity. Now, the factors are land conversion. If there's a process of land conversion, then somehow it has a adverse impact on the climate. It has a adverse impact on the habitat, and due to habitat loss, there's a great threat to the biodiversity. Greater climate change and global warming. Now, due to change in the climate or due to change in the temperature of the earth, might be again there's an impact on the habitat, and due to which many species they get impacted. So again they create the threats for the biodiversity. And pollution. In the previous lecture we have studied the land pollution, the soil pollution, and the water pollution. All the three types of the pollution, directly or indirectly, they can have impact on the species, they can have the impact on the habitat, they can have the impact on the human health. Then exploitation of the natural resources. If we are continuously doing the poaching of the animals we have studied in the previous lecture, if we are continuously using the forest for the economic use, then somehow we are exploiting the natural resources. And if we are doing the continuous consumption of these natural resources, then might be we are creating the endangered for few other species, and then they create the threats to biodiversity. Interference of the ecosystem by alien species, habitat destruction, and genetic manipulation of the species. So these are the particular factors which are responsible for creating the threats to biodiversity. Now, the previous causes, they are the main causes like land conversion, greater climatic change, global warming, pollution, exploitation of natural resources, interference of ecosystem by alien species, habitat destruction, and genetic manipulation of species. These are the factors which have direct impact on the threats to biodiversity. But there are few of the factors which are creating a threats to biodiversity in a secondary manner or in indirect manner. So the secondary causes are the population growth, unsustainable pattern of consumption, increased waste production, urban development, and international conflicts. Then comes the mega biodiversity and hotspots. Distribution of biological species across the world differ with some area having full of life and other without life. However, the majority of region falls in between. Megadiversity zone can be defined as the regions with large number of species in an ecosystem. Hotspot concept was developed by Norman Myers in 1988. These are the richest and the most threatened reservoirs of plant and animal life on Earth. There are 34 hotspots for conservation of biodiversity and has been identified worldwide, including 4 Indian hotspots. Conservation of biodiversity. Conservation is defined as management of human use of biosphere so that it may yield sustainable benefit to the present generation while maintaining its potential to meet the needs and aspiration of prosperity. There are two basic strategies of biodiversity conservation. In situ conservation, that is on-site conservation, or we can say the conservation in the natural habitat. Even we have discussed this concept in previous lecture. Ex situ conservation, that is the off-site, means other places rather than their natural habitat. That comes under the ex situ conservation. In situ conservation. Conservation of organism in its natural home through protection of a group of typical ecosystem by a network of protected areas. Biospherism, sacred forests, and sacred lakes. Protected areas such as national parks and wildlife sanctuaries are areas of land and or sea especially dedicated to protection and maintenance of biological diversity and of natural and associated cultural resources. There are more than 37,000 protected areas around the world, while India has 612 protected areas, 99 national parks, and 513 wildlife sanctuaries. Then biosphere reserves are a special category of protected areas of land and or coastal environments wherein people are an integral component of the system. A biosphere reserve consists of core, buffer, and transition zone. The core zone comprises an undisturbed and legally protected ecosystem. The buffer zone surrounds the core area and is managed to accommodate a greater variety of resource use strategies, research, and educational activity. So over here with the help of diagram, you can see at the center there is a core zone which is surrounded by the buffer zone and the outermost layer that is the transition zone. The transition zone is the outermost part of the biosphere reserve, is an area of active cooperation between reserve management and local people, wherein activities like settlements, cropping, forestry, and recreation and other economic uses continues in harmony with conservation goals. There are 408 biosphere reserves located in different countries, out of which 13 biosphere reserves located in India. The structure of model biosphere reserves. The dark green area, you can see that is the core area. Then the light green area, which surrounds the core area, that is the buffer area. Then the gray color area, you can see that is the transition area. Then the home settlements or human settlements. Then R stands for the research area. E stands for the education and the training for providing the expertise. And T for the tourism and recreational. So these are the following demarcated area which are particularly assigned for the enhancement of the biosphere reserve and for the conservation of biodiversity. Now comes the XC2 conservation. Conservation of organisms in botanical garden, zoo, conservation stands gene, pollen, seed, seedling, tissue culture, and DNA banks. There are more than 1,500 botanical gardens in the world containing more than 80,000 species, and many of these now have seed banks, tissue culture facilities, and other ex situ technologies. Similarly, there are more than 800 professionally managed zoos around the world, with about 3,000 species of mammals, birds, reptiles, and amphibians. Many of these zoos have well developed captive breeding programs. Plants and animals conserved in botanical gardens, arboreta, zoo, and aquarium can be used to restore degraded land, reintroduce species into wild, and restock depleted population. Biodiversity conservation in India. India is known for its rich heritage of biodiversity. It is one of the 17 mega diverse countries in the world with 7 to 8% of the world's recorded plant and animal species. Amongst the existing biota, 91,307 species of animals, 2,557 protista, 12,470 general invertebrates, 69,903 arthropods, 4,994 vertebrates, and 45,500 species of plants, as well as 5,650 microbial species have been documented in India. About 5,150 plant species and 1,837 animal species are endemic to India. Its cultural and traditional diversity includes over 550 tribal communities of 227 traditional groups spread over 5,000 forested villages. Some important techniques used in ex situ conservation are dealt here. Under that, the first that is a captive breeding. This is the most important mean for ex situ conservation. The captive breeding is one of the important strategies used by both government and non-government organizations. Some important techniques used in ex situ conservation are dealt here. Under that, the first is the captive breeding. The captive breeding is one of the important strategies used by both government and non-government organizations. Captive breeding, captive breeding programs of endangered and threatened species have become familiar programs that strive to preserve biodiversity and species survival plans such as cheetah. The second is the embryo storage and transfer technology. The techniques for embryo transfer and artificial insemination, which have been developed for laboratory animals and farm animals, are potentially very useful for improving here their productive potential of captive populations of endangered species. These kind of techniques have been worked out mainly for mammals. The third is artificial insemination. Artificial insemination is another technology that may be useful. A sperm can be frozen and used later or transferred to another breeding facility to increase genetic diversity. Sometimes the sperm can be added to the eggs in a dish and fertilization will occur. In other cases, for example, horse, the sperm has to be injected into the egg. A few years ago, the black-footed ferret was down to 6 individuals, but artificial insemination has now been used to produce 16 kittens. Elephant and cheetah have conceived, and a cheetah cub has been born following artificial insemination. Elephants have not bred naturally in captivity, so this method may be useful simply to make captive breeding possible. Then comes the somatic cell cloning. Somatic cell cloning holds some promise for propagating from one or few survivors of an almost extinct species. This was first done with domestic sheep at the Roslin Institute in Edinburgh from University of Virginia, but has since been done with other mammals. It has already been used to rescue a rare breed of cattle that had been reduced to a single old female lady and some frozen sperm. Granulosa cells, somatic cells in the ovary from lady were fused with the enucleated egg, lacking DNA from a different breed, and the resulting eggs were implanted into an Angus cow, a common breed. The first cow born from these cells is genetically identical to Lady, as expected, although her markings were slightly different. Then comes the fostering. Many eggs laying animals, that is, birds and reptiles, are capable of producing many more eggs than they can rear. This raises the possibility of collecting the extra egg and hatching and rearing the animals in captivity with a foster parent, then using them to settlement wild populations. It has worked extremely well with some birds, particularly the peregrine falcon, which is now doing so well that the fostering program is being phased out. Rearing of whooping cranes has also been successful, and the species recovered from a population of 21 birds in 1941 to over 300 in 1996. So, till, till here, we have completed various methods of ex situ in situ and ex situ conservation. Now, the remaining part we will cover in next lecture.